Hi there, my name is Atley Muila, the application specialist for genomic services here at Ingqaba Biotech. After going through all the projects and videos submitted for Ingqaba Biotech Africa BP Genome Challenge, I am pleased to let you know that we finally have a selection of projects and videos that you, the Africa BP community, will have to vote for an opportunity to get those projects sequenced for free on our PacBioSequel 2E system here at Ingqaba Biotech. How did we get to Ingqaba Biotech Africa BP Genome Challenge? One might ask. Well, back in 2021, Ingqaba Biotech launched the inaugural Ingqaba Biotech Genome Challenge 2021, where researchers across Africa had to submit their project proposal as well as the motivational video for an opportunity to get their species of interest sequenced on our PacBioSequel 2E system. Now, in recognition of Ingqaba Biotech's 20th anniversary in 2022 and the partnership with Africa BP, the 2022 version of the Genome Challenge was dedicated to the Africa BP community in an effort to assist the Africa BP to advance their goal of sequencing species of great importance on the continent. Now back to the selection of the videos. The following project videos were selected based on the following criteria. The species must have scientific relevance, economic value, biodiversity and conservation, and must be of cultural significance. Please watch the following project video until the end to find out how you vote for your favorite video. Enjoy.
Hello, my name is Sabah Ibrahim. I am a researcher at the National University Biomedical Research Institute, and I am proposing to study the whole genome sequence of Bagras bayad, and it will be the first study of its kind in Sudan to study the genomic diversity of this fish. Bagras bayad is found all across the River Nile, and it is considered as a high-quality, economically valuable fish in Sudan. Besides, it represents a candidate catfish breed for aqua farming development. Aquaculture is an excellent anti-poverty solution in Sudan, as it assists in food security and creating employment opportunities, especially in rural areas. In Sudan, fish farming production is still low when compared to the worldwide share. Whole genome sequencing can enhance in diverse farming environment by selective breeding programs, fish health, and monitoring migration, which leads to cutting costs, increasing efficiency, and improving sustainability. Thank you.
Do you like bread, cake, chips, and rice? They are all product of crops, but with climate change, we risk losing them in the future. We are the Plant Stress Lab of University of Cape Town. Our research focuses on the way in which plants survive extreme drought stress.
One of the plants we study is Anemia capharum, an amazing fern that grows right here in the Western Cape. Although a lot of ferns are water-wise, Anemia is the only one that is desiccation tolerant, meaning it can dry completely and come back to life when rewatered.
It does this through a complex series of mechanisms which freezes its metabolism, protecting its cell structure until rehydration. This amazing fern has another incredible feature. It is the only known seasonally desiccation tolerant plant. In the winter, it loses its desiccation tolerance, only to regain it again in summer. This makes it an ideal model to study the induction of desiccation tolerance.
Using a systems biology approach, our lab has been researching this unique fern for over 10 years. However, its genome is the missing link. Sequencing the genome of this extraordinary fern would help us decode the induction of desiccation tolerance, which could pave the way for improving crops in terms of surviving drought. Assembling the genome after sequencing would promise to be a challenge, but we have brought together an expert team from Europe and America to collaborate with us.
With this team, we can make the most of the sequencing data, so please vote for our project and save our comfort food.
My team and I propose to conduct a whole genome sequencing of Tapenes pars species, the horsefly. Approximately 4,300 species have been described worldwide, and from these, 700 species are said to occur in Africa. When they are present in high densities, they cause a significant economic impact on tourism and livestock production. They also have been identified as vectors of animal disease agents, transmitting bacteria, viruses, as well as parasites. They also contribute into the floral diversity in that the tapenid flies, male species, serve as pollinators, which ensures biodiversity and species richness of flowering plants. Research indicates that the tapenids are a neglected subject to an extent that there isn't an available whole genome sequence of the tapenid flies, which is why my team and I aim to pioneer the tapenid whole genome sequencing. In doing that, we will answer genetic diversity related questions and thus contributing to a successful pest control in Africa and the world at large. Thank you.
Hi, I'm Victor Zemmour. I'm presenting our project. The theme of the project is in silico characterization and phylogenetic study of soil worm isolated from Algeria. Soil worm sequencing will help us to confirm the morphological identification. Secondly, to do in silico characterization of the resistance and growth hormonal genes. And thirdly, the study of the phylogenetic and biodiversity. The interests of the project are the better knowledge of this organism to improve the plant health, will increase the agriculture yield, and finally, socio-economic impact to fight famine in Africa by increasing nutritional quality, thus the human health.
I am Chinye Choma Isosu, and I would like to sequence this plant, Trichulia africana. It is an economic and nutritious crop found in West and Central Africa. It is also known as African breadfruit and Ookwa in certain parts of Nigeria where it's a delicacy. It's an endangered and neglected crop which is threatened due to urbanization and deforestation. There's a depth of molecular data of the crop in the genetic database. Therefore, whole genome sequencing will help provide information needed for its improvement or even to address some of the challenges facing the crop by determining the genetic variability of the varieties or characterizing the varieties, and also to identify molecular markers associated with genes of some agronomic traits. This will in turn make the fruit available all year round to meet the demands of the consumers at an affordable price, and at the same time improve the economic activities in relation to the plants. I want you to vote for Trichulia africana. I want you to vote for Trichulia africana. Thank you very much.
I guess the next question is, how do you vote? The titles of the project will be in the comment section below, and the project with the most like will automatically be selected as the winner. Don't forget to subscribe to Ingaba Biotech YouTube channel and turn on the notification button so that you don't miss a thing. Get as many people to like your project to stand a good chance. Good luck.